Hello, my beautiful biologist. Welcome to another biology with Miss Judd. Today we get to start our Unit 4, which is on the central dogma of biology. This central dogma is just the process by which the instructions in our DNA are then converted into a functional product, which would be an organism. This was first proposed in 1958 by Francis Crick, who is credited with the discovery of the structure of DNA. The central dogma of molecular biology explains how genetic information is converted to make a functional product called a protein. And then those proteins are put together to make an organism that has different traits and functions. So DNA is the information molecule. It stores the instructions. And then those instructions are stored inside each of your cells. In order to understand the biological function of DNA, we first need to understand its molecular structure. This requires learning the vocabulary for talking about the building blocks of DNA and how these building blocks are assembled to make DNA molecules. The person I think best for teaching us this vocabulary is Miss Judd in the corner. Hi, you guys. Miss Judd in the corner here to talk about Lesson 1 on DNA structure and function. Now, the first question we need to ask is, what is DNA? What are we even talking about? DNA is short for deoxyribonucleic acid. Now, obviously, that's a really long word, lots to say, which is why we just call it D for the deoxy, N for the nucleic, and A for the acid. DNA is the genetic material responsible for all traits in all living organisms. The way we look and act is coded in our DNA. Because this is super important, we keep this safe inside of the nucleus. Remember, that's an organelle in eukaryotic cells. So inside eukaryotic cells, we have our organelle, the nucleus. Inside the nucleus, we have our chromosome. And inside that chromosome is our DNA with that code. DNA is a polymer, meaning it is made up of a bunch of little pieces. Those little pieces are called monomers. The monomer of DNA is called a nucleotide. That's a really important piece. There are 3 parts to a nucleotide. We have our nitrogenous base, we have our phosphate group, and then we have our deoxyribose sugar. And you can kind of see how we get the name deoxyribonucleic acid from these 3 parts. In your notes, please draw this image and label the phosphate group, which is that little circle, the deoxyribose sugar, and then the nitrogenous base. Now we can see how this nucleotide links together to make the DNA molecule shape here. There are little pieces that link together. So this is just one piece, the monomer, making up the polymer DNA. Our nitrogenous base is super important because that is where the code is held. We have 4 different bases: adenine, guanine, cytosine, and thymine. Those 4 bases are in different orders for every single organism. So the differences between us and our frogs is because of these nitrogenous bases. These are super important because this is where that code exists between our adenine, thymine, cytosine, and guanine. Now, how these nitrogenous bases on DNA line up is actually on purpose. It's called base pairing. So the nitrogenous bases on one side of DNA are complementary to the other side. They are held together by hydrogen bonds, which are actually a little weak. These nitrogenous bases were found by this guy named Chargaff. He mixed some DNA together with an acid so those nucleotides would separate out. When a piece of paper was placed in the mixture, the nucleotides all traveled up the paper, but at different rates because they're different sizes. If you look, adenine is bigger than thymine, and guanine is bigger than cytosine. Because of this, he noticed that there was an equal amount of adenine to the amount of thymine. And there's an equal amount of cytosine to the guanine. And what he came up with, this rule, that adenine must always bind with thymine, and cytosine must always pair up with guanine. So this is a really important rule. You might want to draw 5 stars around this. A goes with T, C goes with G, always and forever. You will always find A with T and C with G. So if I have 15 A's, then I must have 15 T's because they're always together. If I have 30 G's, then I must have 30 C's because they're always together. This creates our DNA shape. It is a double-stranded molecule. Purines, which are adenine and guanine, are larger. Pyrimidines, such as thymine and cytosine, are smaller. So when they're paired correctly, they always stay the same width. It's uniform. If you look here, purine with a purine, too wide, too big. Pyrimidine with a pyrimidine, too narrow, not good enough. But if you've hit a purine with a pyrimidine, if A goes with T and G goes with C, it's the same width all the way down this molecule, which gives us our DNA shape held together by hydrogen bonds. The sugar and phosphate are on the outside, the deoxyribose sugar and the phosphate. This creates a super strong backbone with the weaker nitrogenous bases on the inside. Remember, our code is in those bases. So we want to keep that code safe. So we have our strong backbone with the sugar and the phosphate with our code on the inside. And then DNA twists and build a protective shell around that code. And that's why it's called a double helix or a twisted ladder shape. Now, our DNA is super important. As I said, it's the blueprint for all the different proteins in our body. It creates our genes and the coding segment that is specific recipe for a specific protein, which gives us our specific traits, which is why we want to make sure that we understand its structure so that it can actually do its job. All right, back to you, Miss Judd. Oh my goodness, he caught me. That was amazing. Thank you so much, Miss Judd in the corner. I feel like I have a good foundation so that when we start working with this DNA, I know exactly what's going on. Make sure that you guys complete your notes, do the practice, and be ready to come into class to talk about DNA and its structure and its function. You guys are doing great. I'm so proud of you. Have a great day and make good choices.